Brooks. I'll be giving a presentation or an informative type speech on dengue virus. Before we get started, okay, Randall Brooks, Alan, one of my first audience members. All right, over here you have Tony Womble. And you have AKA the Nathan. All right, we'll go ahead and get started on this. What is dengue virus? As you can tell, a virus. So looking at this first slide right here, I put on it's kind of what a mature virus looks like and an immature. This right here is mature, being like has that tall smooth edge. Right here is the immature one, has the spikes. So the actual infectious one, as we go through here, keep this in mind, is this actual mature one. So basically, these two, looking at this right here, you have a scanning an SEM and TEM. TEM down here, transmission electron microscope gives 3D structure. The scanning electron microscope gives two-dimensional. All right, so some of the things we're going to go ahead and discuss as we go through here. We're going to go ahead and discuss pretty much what is dengue virus, and we're going to go ahead and talk about some geography, some structures, talk about secondary structure, and also going into what the genome looks like. So, and we'll also talk about some basic structure and function. All right, so some taxonomy real quick, where dengue virus comes from. It's a taxonomy when you start looking at biology. Flavidaria, then it goes down into flavivirus. Flavivirus is broken down in two sections. You have ticks and mosquito-borne. Now right here, all the mosquito-borne up here is what we're going to kind of emphasize on. We're looking at primarily, pretty much, some of the, your primary carriers. Aedes aegypti is going to be your primary carrier as far as United States. So we're going to go ahead and look at some, this guy's carries yellow fever, Zika, West Nile, malaria, and Japanese encephalitis. All right, real quick history. We're looking at probably 1992, Japanese Encyclopedia. Talks about just some basic signs and symptoms. Also, you go into 1780. Philadelphia is one of the first emergency outbreaks, is what you see. Pretty much, it's called breakbone fever. That's what they call it. Then also this guy, Albert Sabin. You see him the first time in virology magazines or journal articles where they're able to isolate, identify, see what this thing looks like. All right, here are some basic signs and symptoms of what dengue virus is broken down into three things. You have dengue fever, which you might not know it. You might just think it's a flu. Then you go into, all this stuff's tiered. So the symptoms, once you have dengue fever, it goes into dengue hemorrhagic, then it goes into shock syndrome. So you gotta have one before you have the other. So hemorrhagic fever, that's pretty much what you see some of the pictures. People are bleeding out of their eyeballs. Shock, it's pretty, you gotta be in the hospital, you're pretty much dying at that point. All right, basically this right here, we're looking at pretty much the migratory pattern of the mosquito. So right here, all the yellow areas are all the infected areas. And this is actually coming from the World Health Organization. Once we get in the United States, CDC, Center of Disease Control, where we'll see some of the regional stuff. That'll be my last slide. I'll key on. All right, this is what we're looking at right here is, when we talk about viruses, since they're non-living, but we also, in biochemistry, you think of more living things, as far as chemistry, study of matter, biology, study of life. But right here, you can't look at the actual virus, at the actual primary structure, like adenine, guanine, because things like all these viruses are about 70% alike, but the only way you can identify it is through serology, through the blood. And we'll get into actual what we're looking at to see how we look at that as far as those assays. So right here, 4 different serotypes. This fifth one comes in play, 2014, discovered by Dr. Mustafa, looking at his journal article on virology. So I bring this up, this is probably an upcoming thing that you'll see in the United States in the next 10 years. All right, now we look at some structure, the actual gene. All right, this gene, the genome has 10 genes. So when you see this capsule, membrane, envelope, so think of it as your skin, your layers of skin. Capsule, your first layer of skin with contact. Envelope, if you get secondary degree burn. It's your membrane. It's also broken into a PR membrane, which we'll get to the next slide. You'll see that. And you have envelope. Now all these NS 1 through 5 are non-structural proteins, which this comes into replication type mechanisms. And here's untranslated regions up here. All right, so right here we're looking at the basic structure. So we kind of went through what dengue virus is, and we kind of went through where it's going to be located, where you see the locations at. Now we're talking about structure. So it kind of broke this down as far as, here's your primary structure. We'll see what the actual dengue virus looks like. We'll have a zoomed in thing, but we'll see what it looks like. And here's all the non-sequence. Like, there's not a structure for it, but when you look at it, there's a way of testing to see what the stuff is. So once you get a capsule, here's your membrane. It's broken down to a pre-membrane, envelope, and this right here is just showing, here's structures. I don't have primary structure shown on here, but I have secondary. Secondary structure is alpha helixes right here, and beta plated sheets are secondary structures as well. So two major things when you look at secondary structure. Basically, it's just showing each one of these things have different structure, different function, what these structures play into. But remember, all the dengue viruses, all serotypes, all 5 of them have this type of motif or domain and secondary structure. So, yeah, that's all I want to say right there. All right, so we're looking at the actual envelope protein. This is where structures kind of meet function. So right here we have the immature, which is inside the cell, has the spikes, looks really, really aggressive. If you kind of remember back on that first slide what we discussed, kind of what you have the smooth outside, then here you have like, which is going to be this side over here. But this immature one is actually a pH driven, so all that's inside the cell. But once it goes outside the cell, pH driven, it goes mature, it has that smooth shape, and that's what's actually going to bind to receptors on the cell surface, and that'd be the mode of infection. This right here is just showing, this will be one of the ways. I'm not going to go discuss this in this presentation, but this is actually how the thing actually binds, forms a tunnel, pulls out, and will inject its RNA, positive sense RNA, which means it's only one directional when it comes to replication. All right, here's what the mature one looks like. So here you can kind of see like the outer protein or the envelope protein. Here you kind of notice it makes an isohedral shape. When you look at as far as X-ray type imagery, we start looking as far as getting down there. That's a key indicator showing it's a dengue virus. All right, next one is the immature. And you tell this is what the pH driven one I think kind of looks like it's going to start forming spikes. So pH driven conformational change is what you need to get from this. All right, so here we go. We're starting to get into function. Now we kind of talked about the genome. There's 10 different genes. So basically, this thing it actually introduces itself into the ER, and all these little points, anchor points. These are single-ase regions. This is just another important region as far as when signaling has occurred. And here is where some of the replication is going to happen in the cytoplasmic side, not the ER side. So basically, this thing's going to go up the ER, shut down cell machinery as far as replication. It's going to make its own replication, but at the same time, it's misfolding certain proteins. So autophagy is one of the ways that this thing's going to go ahead and perform cell death. So this thing gets in here, it'll gum everything up. Different ways that this stuff can go ahead and shut down a cell, kill a cell. It interrupts the calcium uptake and just calcium moving throughout the cell. Also, you have these phosphorylation events. All these are different pathways, just different ways of shutting down the cell or destroying the cell. Then you have this unfolded protein regulation, which is misfolded proteins. So stuff just gets in the way, clogs everything up, does its bioreplication within the cell, shuts down the cell, destroys the cell. At the same point, the advantage for destroying the cell is there's no immunological response. Last slide right here, we kind of talked about geography. Right here, Aedes aegypti, here's your primary carrier. But Aedes albopictus is another guy that can carry all those, like malaria, Zika, West Nile. It can carry that same thing. So if you look at the regions, temperature regions, and it's seasonal. So I kind of want to make this a point, is why I'm interested in it. You see these mosquitoes flying around everywhere. It's just going to be a matter of time before you see more outbreaks or more cases of this when you see dengue virus. So we kind of went over what the Zika virus is, positive stand RNA, non-living, the structure. Remember, 10 genes make up the genome. Kind of briefed over the replication process that's going to happen in the ER. Gums everything up, shuts everything down, autophagy, destroys everything. Questions, guys? Questions, Ella?
No.
Tony?
No questions.
Okay. Randall Brooks, this is kind of what we discussed. That's why it's important to me, and I hope you enjoyed this discussion about dengue virus. Cut.